Unifying themes, can you even think of all the unifying themes? All right, it's not going to be on the exam, right? So what is this this one here on the, that's your left side, what is this representative of? What? Yeah, but which unifying theme? Yes, I know it's DNA. Yes, correct, it is DNA, but that's not the question. The question is, which unifying theme does it represent?
This? Okay, I was just gonna ask you. I know. Okay. Okay.
Can we think of any others? Right, from chapter 1, 5 unifying themes in biology. Right, one of them is organization, right? Cells have organelles, tissues are made out of cells, organs are made out of tissues, right? Those are different levels of organization in the biological hierarchy, right? The transfer of energy and matter, right? So we take sunlight energy, plants take sunlight energy, turn it into chemical energy. We take that chemical energy, right, use that, et cetera, et cetera. Right? Interactions, right? We have interactions at several different levels. So, for example, organisms within a population can interact with each other. We can also have, we saw several interactions last time we met between, for example, a ligand and a receptor. Those are all examples of interactions, right? Information, right? One of the unifying themes of biology is that information, right, is passed on from one generation to the next via DNA, right? So this is representative of the information one, and then the last one is evolution. Those are your 5 unifying themes of biology, right? So a picture of DNA would be representative of information because that's how we transmit information from one generation to the next. What about that other one? Which one of these 5 unifying themes? Yes, Avanish?
Evolution.
There we go. Specifically, what do we see there?
Wings. Wings.
Okay, specifically what type of evolution do we see there?
Wings.
Different wings suited for different environments and different body sizes.
Yeah, this is, yeah, there we go, that's what I'm looking for. Convergent evolution, right? We have several different species there from several different organizational parts of life, but they have all independently developed wings because that is functional for them, right? So yes, convergent evolution is what that one is representing.
How many evolutions are there? How many types?
Several. All right, so that's one example of what I might ask you, right? Give you an image, ask you which of the unifying themes. Of course, it's helpful if you know what the unifying themes are. How about emergent properties? What are emergent properties? Can we just define that idea?
What was the question?
The second part here, right? Emergent properties are characteristic of biological organization. What are emergent properties? They become apparent as we move up in that organizational hierarchy, right? So as we go from organelles to cells, right? The individual organelles don't have the same properties, right, as the cell when you put all that together. Right? The mitochondria can do one thing, right? A chloroplast can do one thing, right? But when you put those inside a eukaryotic cell, right, then we have additional properties that are possible that weren't possible when we just had the organelle. Right? This might be the basis of a theory, right, by which mitochondria and chloroplast are considered or thought to have once upon a time been independent and then incorporated into your eukaryotic cells. Anybody remember the name of that theory? What would we call it when a symbiotic organism might be absorbed into another organism?
Endocytosis.
That's another word. The endo part is right. The s part is right. There we go. Endosymbiont theory, right? The endosymbiont theory, right, kind of builds on this idea of emergent properties, right? That early eukaryotic cells did not have the capability of fully breaking down molecules until they formed a symbiotic relationship with mitochondria, which could do that for them. And so now they have a new property that they didn't have before. All right, domains of life, examples of each. From which domain of life do you think these organisms came? It's a decent guess, right? Archaea is on the table. That's not actually what this one is, but it's a decent guess. Is it eukarya? Are those eukaryotic cells? Bacteria, right? Our three domains of life are bacteria, eukarya, and archaea. Archaea we really only see in extremophile conditions, so like thermal vents, bottom of the ocean, things like that, right? Bacteria, right, are prokaryotic organisms, as are archaea, and then we have all the eukaryotic organisms in eukarya. Right? If you looked at the slide of bacteria when we did the microscopy lab, then you saw something that looked like this, and you'll recognize those as bacteria. Let's see here. What about this other one over here? Which domain of life do we think this is? Why do we think it's eukarya? You can see organelles, right? That's what distinguishes eukarya, right? Or the cells have organelles. Very good. Within bacteria and archaea, we don't have a whole lot of subdivisions, but within eukarya, we do have subdivisions. Can anybody name the first level of organization below eukarya? How are all eukaryotic organisms initially organized? Into kingdoms. Right? So among all the eukaryotic organisms specifically, right, we have Animalia, we have Plantae, we have fungi, and we have Protista. Right? Those are the four kingdoms that we divide eukaryotic organisms into. Anybody know what comes after kingdoms? Phylums, and so on and so forth. What's an example of unity among diverse organisms? Any example at all. What unifies this eukaryotic cell with one of these bacterial cells? Is there anything that unifies them? Anything they have in common at all? Yeah, they both have cell membranes, right? They both have genetic information, right? One of them's in a nucleus and one of them's not, right? One of them's embedded in the chromosomes and one of them's typically just one big circle, but they do both have genetic information, right? So that would be an example of unity among diverse organisms, right? Even though they belong to totally different domains of life, they still share common characteristics. What do we have, right? Is this a tree diagram?
Yeah.
The answer is yes. That is what a tree diagram looks like. According to this tree diagram, which two domains of life are most closely related? How do we know that? Yeah, right, they both branch off of the same branch, right? Whereas this one branched off earlier. Kind of surprising, right? Once upon a time you thought bacteria and archaea were more closely related. Turns out they're not, right? So yes, according to this tree diagram, archaea and eukaryota are more closely related. And so if you understand that basic concept, right, then if I say gave you three organisms, right, organism A, B, and C, and asked you to draw a simple tree diagram indicating that A and B were more closely related than the third category C, then I would expect you to be able to draw something along the lines of A, B, C. Right? That would be a simple tree diagram showing A and B more closely related than the third one, C. That's all we're looking for there. All right, interpreting graphs and drawing conclusions. We didn't spend a lot of time on this in lecture, but this diagram comes directly from your book, so hopefully you looked at it. What is the independent variable on this diagram?
Half-lives.
Yes, half-lives. Sorry, I have to think about that for a second. Yes. The independent variable, right, is half-lives, right? That's not going to change, right? It's independent from the data, no matter what data you get, right? That axis is going to be the same. Right? That means the dependent variable must be the y-axis, right? Meaning that it's dependent on the experimental results, right? We don't know what this is going to be, right? Until we actually do the experiment and we have data to put on there. That's why it's dependent on doing the experiment. What fraction of isotope do we have remaining after, what does it say, 7 half-lives?
Yes.
Right, that's not 7.5 lives, that's 7 half-lives. Could have drawn that better, right? But how do we even figure that out? What fraction is remaining after 1 half-life?
Half.
Yeah. How do we write that in scientific notation?
0. 1.
There we go. How about after 2 half-lives? How much is left?
Wouldn't it be positive 1, not negative 1?
Five times.
Oh yeah, right. I just did that all kinds of wrong. There we go. How about that? You like that better? There we go. Right. So I might ask you to do something similar to this, right? So a quarter, right, would be 2.5 times 10 to the negative 1, so on and so forth by the time we get here, which is going to be 0.078, right, which equals 7.8 times 10 to the negative, 12? Right, so what fraction of isotope remains after 7 half-lives? Right, 0.078 or 7.8 times 10 to the negative 2. We all know scientific notation, right? We could do this? Okay, good. If the isotope is carbon-14 with a half-life of 5730 years, then according to this graph, what is the useful limit of carbon dating?
2.5.
And this graph shows that it gets to 0% remaining by approximately 10 half-lives. If each half-life lasts 5730 years, times 10 half-lives equals 57,300 years. All right. Anybody know what carbon-14 decays to? This was in the reading too. I don't know if you came up with it. I didn't say it, but. All right. So what is carbon normally? Yeah, carbon is 12. Right. So if we have carbon-14, what does that mean, right? This normally it has 6 protons and 6 neutrons. All right, if it's carbon-14, then how many neutrons does it have?
8.
Right, equals 8. Right, if one of those neutrons decays, right, then it'll have 6 protons and 7 neutrons. Sorry, I had that the other way. I had that wrong, don't I? Oh, it decays into a proton, right? Then it has 7 protons. Right, it'll have 7 and 7, and that makes it nitrogen. Thank you. 7 plus 7 is indeed 14. You don't really need to know what it decays into. I was just wondering if you knew. There's another graph in chapter 1, I think, that has like some data related to mice on either a light background or a dark background and whether or not they're likely to get caught. There's another graph that talks about acidification of oceans, talks a little bit about buffering systems, right? Have a look at those, right? Just make sure that if I stick up a graph, no matter what graph that is, you can answer basic information, right, about that graph. What are the 4 elements that make up most of life on Earth? I'm hearing some mumbling. Does anybody have a yes, Avanish? Carbon's 1, good. Somebody else.
Oxygen.
Oxygen's 1, yes.
Nitrogen.
Hydrogen and nitrogen, yes.
Hydrogen.
There we go, right? So those are the 4 elements that make up 96% of all living matter on Earth. Can we name 2 weak interactions crucial to life? Hydrogen bonding is one, yes? What's another one? Is covalent bonding a weak interaction or a strong interaction? Yeah, van der Waals. There we go, right? Van der Waals, hydrogen bonding, right? The two stronger interactions, right, include covalent and ionic bonds. Ionic bonds aren't particularly relevant to life, though, because of what reason? How come we don't talk about ionic bonds very often when we're talking about biology? The ion thing, right? Yeah, right, life on Earth, right, is a water environment. In water, ionic bonds, right, dissociate and you just get ions, right? So we don't see ionic bonds in living things because they can't be bonded, right, because they're ions. Lots of covalent bonds though, lots of weak interactions. Which of the following A through F is an isotope? A is an isotope. C is an isotope. F is an isotope. All very good, right? So, right, carbon's normally 12, so that's the isotope. Oxygen's normally 16, so that's the isotope. Hydrogen's normally 1. Okay, good.
We're not gonna get any other.
That's about as chemistry as it's going to get for this class. It's not a chemistry class, but if we can talk about chemistry as relevant to biology, then it's fair game, right? So we don't want to get too deep into it, but we want to make sure we understand the basic principles anyway. Okay, could we draw two water molecules and show the hydrogen bonds? Everybody feels confident about that? Yeah. Then I won't do it, because everybody said yes. Oh, now we're not so confident. If you're not confident, be sure to say no, so I know you're not confident. If all I hear is yes, then I'm going to move on. All right, what do I need for a water molecule?
2 hydrogens.
There we go, 2 hydrogens and an oxygen. What do I need for my other water molecule? 2 more hydrogens and an oxygen. Where would the hydrogen bond be? Right, because we have a slight positive and a slight negative. Right, we have a slight negative and a slight positive, right? And so the negative, the positive regions of these molecules, right? So there's our hydrogen bond. That's all I'm looking for there. What are 4 properties of water that result from hydrogen bonding? What's one of them? Adhesion.
Cohesion.
Yeah, cohesion's the same thing. Here we go. I'll write them together for you. There's one. Adhesion, cohesion. Don't write them as separate things. They are not different from each other. It's the same property, right? Adhesion, right, is water molecules sticking to other polar things. Cohesion is water molecules sticking to themselves, but for the same reason, right? What's another one? Yeah, right, so, right, why is water able to absorb a lot of energy without changing temperature? Well, it's hydrogen bonding, right? That's the answer to all these questions, right? But hydrogen bonding, right, means that it takes more energy to wiggle these apart because they're attracted to each other. If they weren't attracted to each other, it wouldn't take as much energy, right? Because we don't have to overcome that hydrogen bond, right? So, yes, you could say specific heat, right? You could say heat sink, anything that's related to temperature. What's another one? Yes. Yeah. What if we put temperature up? What if temperature went down? Then it would freeze, then it would float, right? Right? Ice floating is a result of hydrogen bonding because why? Right? Since these water molecules are going to orient to each other to make those hydrogen bonds happen, they're not just randomly getting closer together, right? They're going to be ordered, right? All the hydrogens are going to point that way, all the oxygens will point that direction, right? That's not true of things that aren't attracted to each other. If we have 2 molecules that are not attracted to each other, then as they get closer and closer as the temperature, goes down, right? They'll just have a random association with each other. Right? But since these are not random, right? They orient themselves, right, according to that hydrogen bond. And so it's not random. In fact, it's very ordered. And when we have ordered structure, right, that's a crystal, and the crystal is going to take up more space than if they just randomly packed in really close to each other. Right, so ice floats rather than sinking. What's the fourth one?
It's a solvent, universal solvent.
Yeah, is it really universal though? Of course not. But water is often referred to as a universal solvent because water is polar, as are a lot of things that might be dissolved in water. Turns out on Earth, we have lots of polar things. Okay, good. What is that thing? That's an amino acid, right? The general structure of an amino acid. Which carbon is the alpha carbon? The middle one. Very good. What does the R group represent? Yeah, right? 20 different possible side chains, 20 plus different side chains. What do we call that thing on the right side of the amino acid? Yeah, what do we call it on the left side? An amino group, right? Very good. Right. How would that amino acid look, right, when pH is low? Would it change or would it look the same? How would it change? Where should I put the extra hydrogen, right? pH is low, so there are lots of protons in the solution. One of those protons is going to stick onto this amino acid. Where should I stick it?
On the H2O, dehydration.
Did you say right here? Is that what you meant? It's already, it's double bonded. It can't accept anymore. There's already one here. No. The amino group. Right, when pH is low, right, that is the most number of hydrogens that that can hold, and so there it is, right? What if pH goes back to neutral? Right, then that proton will come back off. What if pH is high? Which of those protons is going to come off? Yeah, right. That one will leave. Right? So when there are very few protons, right, that one will come off, right? As the pH goes back down and there are more protons, first this one will add back on, and then additionally we can add one right here as well. That means that amino acids can act as what? Buffers, right? So amino acid has the ability, right, to both donate and accept protons, therefore it is an example of a buffer. Good. What if it wasn't an amino acid? What if I chose a different molecule, a totally different molecule? Right? Would you still understand the concept? Right? The ability to either donate or absorb a proton. Right? And so what we're looking for then, right? What if I drew a, I don't know. Which functional group is most likely to release a proton? Yeah, right, or just the hydroxyl itself, right? That's a really obvious choice, right? So if you're looking at a more complex molecule than this, right, and I've told you that it can act as a buffer, right, look for a hydroxyl, right? That's the most likely place, right? Because oxygen is very electronegative, right? So the hydrogen is kind of getting the short end of that stick, right? And so if you see, right, the hydrogen stuck onto an oxygen somewhere, that hydrogen is a very likely choice to be the one that's going to be donated. Oxygen don't care. Oxygen doesn't even want that proton around. Oxygen has what it needs. The oxygen got its electron. It's happy. It doesn't give a crap about that proton. All right. Which functional group is this? Hydroxyl. There we go. Right, which we could also draw. Yeah, so this is a hydroxyl. Right, which we could also just draw like that. What's this one?
No, that's methyl.
There we go. This is a methyl. Right, which again we could just draw like that. Same thing. What's the next one? Carbonyl. Right. that one, right, one of the R groups is an OH, so this one specifically is a carboxyl, which we typically draw like that. All right, the next one, what'd you say? There we go, which
Something I mean.
we very often draw like that. What's the last one? It's a phosphate, which we very often draw as PO4, -2, I think. What's a property of a hydroxyl group? Yeah, right. Hydroxyls are polar. What about methyls? Yeah, non-polar. Carbonyl? Got a carboxyl. It actually has a charge, right? A negative charge. Can anyway. All right. What about the amino? Positive charge. What about the phosphates? Negative charge. Good. Y'all are doing great.
I'm gonna need that folder and not pull everything. Pull back out.
All right, so what are some examples of dehydration reactions? It's a very generic answer, but yes, yes. Can we name a specific type of bond that would be formed through a dehydration reaction? So ester is one. Glycosidic is one. That's a G, by the way. I think I heard somebody whisper dipeptide. All right, very good. What if I drew an amino acid? All right, so let me draw. Should I draw it higher? I can draw it higher.
You're already complicated with the filter, how to push it.
All right, let's add those two amino acids together. What do I want to circle on this image? Where's my water coming from since it's a dehydration reaction? Okay, so there's an OH and there's an H. Those will leave together as a water molecule, right? That's why it's a dehydration reaction, because it's losing water in the bond formation. Right, that will allow this nitrogen then, instead, right, to bond to that carbon, right? That's our dipeptide bonds, right? While we're looking at this, can we identify the C terminal of this dipeptide? On the right side, right? Because that's where we have a carboxyl group, right? C for carboxyl. All right, so where's the N terminus? Over here, right? N for amino. N for the nitrogen in the amino group, right? C for carboxyl, right? So no matter how long this peptide is, there's always going to be a free carboxyl at one end and a free amino at the other end, because they all bond to each other exactly the same way, right? So we always have a, whether it be a dipeptide, a tripeptide, a very long peptide, if that peptide folds into a complete protein, right, it's still gonna have one free end that has a carboxyl and one free end that has an amino, right? So every protein has an N terminus and a C terminus. Okay. Could you do the same thing for two glucose molecules? Why not?
Oh my god.
You better take a picture, you better take it fast, I need this board. Or you could write it down yourself. Actually writing it down, like physically taking notes, is actually more effective for learning than snapping a picture, which you then just passively look at later. It's fine though, I won't get on that soapbox right now. Does anybody want to come up here and draw a glucose molecule? What should I draw right here? An oxygen, right? What should I draw up here? There we go. What's the first carbon? What's carbon number 1? On the right or at the top? Yeah. All right, so that's carbon 1, 2, 3, 4, 5, and 6. Right. I'll draw another glucose molecule. All right, so this is 4, 5, 6 again, 3, 2, 1. All right. What do you imagine must be right here? H and an OH, right over here. Right, an OH, another H. Right, and so we can see how that would leave as a water molecule, forming what type of glycosidic bond? Glycosidic linkage, if you want to call it that. Because there are more than one type, right? Since it's between carbon 1 and 4, what do you think?
1,4.
Yeah, it might be a 1,4 glycosidic, right, bond or linkage. Right? So that's 1,4 when we attach two glucose molecules to each other. Right? What would be the difference between a 1,4 and say a 1,6?
What is that? Vertical? What is parallel?
Not quite there yet. I'll ask that question next. All right, where's the 6 carbon? Up outside the ring, right? All right, so if we have a 1,6 linkage, right, that means that we're going to have another glucose molecule up here. Right? What does that mean? What have we created when we do a 1,6 versus a 1,4? We've created a branch point. That branch point is a characteristic of what kind of molecule? When do we have branch points in our string of glucose molecules? Yeah, amylopectin is one. What would be another one? Amylin is another one. Both of those are types of starch. Right, so if we get that 1,6 linkage, we get a branch point. That branch point, right, is characteristic of amylopectin, actually not amylin because amylin is just a spiral, right? But both of those are types of starch. When else might we see a 1,6 linkage, i.e. a branch point? Not just starch, not just amylopectin, also something more relevant to the human body as opposed to plants. Glycogen, there we go, right? So, right, we get a long line of 1,4, right, 1,4, 1,4, 1,4, that gives us a string of glucoses. As soon as we branch off of that 6 carbon, right, now we have either amylopectin or glycogen. Right? What's the difference between amylopectin and glycogen, other than the species in which we find them? Glycogen tends to have more of those branch points. Right? So glycogen is more branched, whereas amylopectin is less branched. Right? What if, Right, what's the difference between these 1,4 linkages in something like amylopectin and the same 1,4 linkages in something like cellulose? One key difference. Yeah, you've both got it, right? So in amylopectin and glycogen, right, we have all the alpha form of glucose, but there's also a beta form of glucose, where essentially, right, this is flip-flopped, right? And so when you link those 1,4 linkages, you'll have one that's this way, and then one that's the other way, and then one that's the next way. And by alternating, right, whether the OH is down or up, we get a long chain, right? Versus with all the OHs pointing up, it tends to kind of spiral, right? So amylin has a long spiraling structure. Cellulose has a very long linear structure. Amylopectin has branch points at the 6 carbon, and glycogen has even more branch points using those 6 carbons. Okay. And then what about for lipids? Are they polymers? Right, these are both polymers, right? The monomer was glucose, right? With the peptide, the monomer was an amino acid, right? What about for triglycerides? Right? Are those polymers? They are not polymers, right? Because we have our 3 carbon, just lost the word, glycerol, 3 carbon glycerol. But then we attach that, right, via oxygen, a long carbon chain. Right, and so since we have, right, a mixture of glycerol as well as fatty acids, right, we can't call either one of those the monomer, right, because they're totally different molecules, right, so it's not a polymer. Right? A polymer requires that we have the same monomer, right, or very similar monomers. They have to at least be the same type of molecule, repeated over and over and over and over again, right? So this is not a polymer because it mixes 2 different types of molecules. But, right, we still have my brain, a dehydration reaction. Right? It's still a dehydration reaction, right? Because prior to putting that on there, right, the head group of a fatty acid is actually a carboxylic acid, right? And the glycerol has a bunch of OHs on it, right? And so, right, we still get a hydrogen and an OH to leave as a water molecule, right, forming a bond there between that oxygen and that carbon, right, so that we have our ester bond in this case for a dehydration reaction. Right? So still formed through a dehydration reaction, right, just not a polymer technically. All right, and then if we took this off of here, and instead we put a phosphate on it, and then to that oxygen we've bound some sort of nitrogen containing molecule. Right, what would we call that? I'm sure you're whispering phospholipid, right? If we take the same structure and we replace one of the fatty acids with a phosphate, right, then we have a phospholipid. And that nitrogen containing thing could be what? Can anybody list one example of what that nitrogen containing addition could be? That's all right, you'll read about it later. It could be something like serine, or choline, or inositol, or ethanolamine, right? All examples, right? So if this is a serine, we call it phosphatidylserine. If this is a choline, we call it phosphatidylcholine, and so on and so forth, right? All membrane phospholipids. Let's see here. Yeah, that's all. All right, what the heck is this? One of them's a glucose. I don't see any branch points, so I don't know if it's starch or not. What's a general term we could use for that? Okay, that's too general. What's a slightly less general? What's a word that starts with poly that we could use here? It's not a polypeptide, it's not amino acids. Yeah, right, glucose is an example of a saccharide. So multiple glucoses would be multiple saccharides, so polysaccharides, right? So what kind of bonds do you see there? Glycosidic bonds, right? Because it's a polysaccharide and they're formed by glycosidic bonds. How many glycosidic bonds do you see there? Yeah, I see almost 6 full glucose molecules, so there are 5 glycosidic linkages there, right? And then what's the monomer? Again, we've already said that, glucose. Right? Do you see the 6 glucose, or the 6 carbon sticking out of the ring here? You recognize that? Right, here we have a hydroxyl, here we have a hydroxyl, right? There's that other oxygen, and then there's a carbon with a hydroxyl on it, right? Here it is sticking down, here it is sticking up, here it is sticking down, there it is sticking up. So what do we think this molecule is then? Polysaccharide known as cellulose, right? Because cellulose is the one where it alternates, one up, one down, one up, one down. What about over there? All right, what do we think this one is? Right, in this case, right, all the 6 carbons are pointing up, right? We don't see any branch points, so that one would be amylin. Right here, right, we see the 1,4 linkage, but we also see the 1,6 linkage, so we have a branch point. All right, and so that one, right, would either be amylopectin or glycogen. We don't see enough of it to know the difference there. Yeah.
Are we gonna see more questions that's about on the left?
These are all example questions that I might put on the exam. They're very similar to the ones that I put on the exam. Yeah, stuff like this. What is that thing? It is a protein, right? What levels of structure to that protein can we see? Right? Which secondary structures do we see? I do see an alpha helix. What else do I see? Yeah, that's it, just an alpha helix, right? What else might I look for, though, for secondary structure? A beta sheet, right? So in this case, I don't see any beta sheets. I only see alpha helices, but those are both examples of secondary structure. What other level of structure can we see? Yeah, we can also see tertiary because we can see the association of those alpha helices with each other. Right? What level of structure can we not see? Yeah, we can't see the primary structure, right? I can't see the individual amino acids, right? And so I can't see that primary structure. What other level of structure can I see? There is one more that I can see. And the colors kind of are the clue that give this away. Not the green color though, the red and the blue is. Right, so we see 2 blue subunits and 2 red subunits, right? So that's quaternary structure, right? Where we associate more than one protein for the final functional protein. Right, so since we have, right, 1, 2, 3, 4 subunits, right, that's 4 different proteins that have to all come together to form that quaternary structure. Right, so we can see secondary alpha helices, we can see tertiary, how those alpha helices are associated together, we can see quaternary, right, the way the different proteins are associated with each other, but we cannot see primary because we can't see any individual amino acids. Finally, what else can we see? We can see one other thing. This is your green. Right? What would green probably indicate? Like this little thing right here, and here, and here, and here. What do you think those might be? We need more information to know if it was a disulfide bridge. What if I told you this protein is an enzyme, right, that is capable of binding something? What would that something be? Yeah, it could be a substrate or more specifically a ligand. Right? Those are both fine terms to use for that. And the area of this protein where it binds its substrate and or ligand would be called what? The active sites, right? So we can see 4 active sites, right, with a substrate or ligand bound. Okay. What determines protein structure? A couple of things. Hydrogen bonding is one really important one.
Interaction between R groups.
Yep, interactions between R groups. Right? And what might one of those interactions be that we look at in lab, for example? What's that? That's actually not a bad answer, right? Because hydrophobicity, right, will tend to turn, right, those towards the interior or the core of the protein, whereas hydrophilic will turn towards the outside, so that's part of it, right? What else could we say with regard to the R groups? What if there was a methionine or a cysteine? What's special about methionine and cysteine? Unique to them and not the other amino acids. If I said the answer was sneaky like a snake. Yeah. I'm glad you got that. Yeah, right, methionine and cysteine both have sulfur, so they can participate, right, in disulfide bridges. There's your disulfide bridge.
Is this silent?
Cysteine and methionine. Yeah, those are your two sulfur-containing amino acids. Okay, I didn't actually have you all do anything with DNA this time. We have the whole third unit of this class is DNA and all that stuff, so we'll save this for then. So don't worry about this. All right, we also didn't really talk in great detail about transcription and translation. Again, we'll talk a lot more about that in Unit 3, so we actually don't need to worry about that for right now. All right, what kind of microscope was used to obtain this image, do you think? Yeah, the one on the far left. Yeah, far left is just a light microscope, right? We're looking at I'll tell you in a second. So we can see the outline of a cell here. All right, we can see the nucleus there. We can see maybe some ribosomes or some vacuoles, kind of the spots inside there. These are actually blood cells, red blood cells around here. Right, and this one, interestingly, is a white blood cell, a type of leukocyte known as a neutrophil, right, which that cell has eaten. But yes, light microscopy. What about the one in the middle? What's that? TEM? We're not going to write TEM on the test, though. There we go. Transmission electron microscopy. How can you tell, right? What's the difference between the middle image and the left image?
2D. It's like a 2D slice.
Well, they're both 2D, so that's not a difference.
Yeah, black and white.
It is black and white, right? Transmission electron microscopy always starts out as black and white. Sometimes it's false colored, right? Sometimes they'll add colors later, so you can't always use that. But, right, the black and white, if you see it, a non-colorized version, that's a very good clue that it's electron microscopy. But what else can we distinguish in the middle image that we cannot distinguish with light microscopy? Or we can distinguish a lot better? Just the organelles in general, right? I mean, I can see the nucleus, and I can see some other hints of nuclei or organelles, but I really can't distinguish the other organelles. Here, right, I can see the nucleus, I can see the nucleolus, I can see the chromatin, and I can see a mitochondrion, right? I can distinguish these organelles a lot better through electron microscopy. Even if you don't know what they are, right, you can still see them better, right, even if you can't identify them. What about the one on the far right? Yeah, that's your scanning electron microscopy. Also, always starts out as black and white, but is very often colorized, right, just to make it prettier. But that one, you're seeing a three-dimensional structure, right? You're seeing the surface of something rather than a slice through it. What's the approximate size range for light microscopy?
One micrometer.
Yeah? What about for electron microscopy? So, in class I said 100 micrometers, but we could probably take light microscopy just to make it a little bit easier, right? We could go from 1 to 1 and then 1 to 1. That makes it a lot easier to remember, right? So light microscopy, right, 1 millimeter down to 1 micrometer, right? Electron microscopy, 1 micrometer down to 1 nanometer. Okay. Yeah. Yeah.
How many questions do you have on the test?
I don't know, about as many as I have slides, I guess. I think there's about 25 questions-ish. 25 to 30, something like that. You want me to share?
Wait, is this gonna be posted on the agenda?
Sure, why not? Yeah, I can put it on campus for you. I don't care. Okay, what do we have in the top left corner there? Here's the top left. What is this? It is a bacteria, which is which domain of life? Or sorry, not, that's already the domain of life, but which type of cell? Is it prokaryotic or eukaryotic? Yeah, right. What can we see in that prokaryotic bacterium? Well, you can see something. Yeah, we see, I don't know if we can actually distinguish the cell membrane, but we can definitely distinguish the cell wall. Right, what else can you see? Yeah, right, you've got all this kind of like grayish stuff in here, right? That's the genetic material known as what? Actually, the word that I'm looking for is escaping me at the moment. I'll think of it in a minute. Yes, but the genetic material. Can you see anything else? Or would you expect anything else?
Ribosomes.
Yeah, ribosomes, right? And so some of those black specks, right, are probably ribosomes as well. That's about all we can distinguish in a prokaryotic cell. No organelles, right? It's all just kind of mismatched inside the cell. What do we think this is? That is a plant cell. They've colored it nicely green for us, just for funsies, right? But what can we make out? Yeah, we have cell walls right in blue here around the corners, right? We have I don't actually see any mitochondria there, although they probably would be present. What do we think these green things are? Those are probably chloroplasts. What do we think this is? Yeah, we got a nucleus with a nucleolus. What do you think these blue things are? Vacuoles, yeah. What kind of instrument do you think was used to produce that image? Yeah. Why do we say electron microscope and not light microscope? I mean, it does look like a thin slice, so TEM, right? And additionally, we can really distinctly make out the organelles, right? Another characteristic of electron microscopy. Right? If this were light microscopy, we could get, one full cell in the image, right? But we wouldn't be able to distinguish the organelles. Right? With electron microscopy, we can distinguish the organelles. What kind of cell do we think this is? Or eukaryotic. It's a eukaryotic cell, right? We can see organelles again. Speaking of organelles, which organelle do we think that is? That is the Golgi apparatus, right? Flat stacks. What do we call those stacks? Begins with cis and ends with turnae. Yeah, the stacks, right, of the Golgi apparatus are called cisternae, right? What would this side of the Golgi apparatus be called? The cis side, yeah, closest to the nucleus and the ER, which makes this surface the trans surface. Very good. And what's the purpose of the Golgi apparatus? Generally to traffic things to the cell membrane, right, after they've been modified in the endoplasmic reticulum. What do we have on the far side there? Mitochondria what? Yeah, this one is, there's only one of them, so it's a mitochondrion, as opposed to multiple mitochondria. What do we call the folds of the inner membrane? Cristae, very good. What do we see surrounding this mitochondrion? Sure looks like rough endoplasmic reticulum to me. Right? Endoplasmic reticulum with lots of little black dots, which are ribosomes. Yeah. And we have a mitochondrion with cristae. We have rough ER over here. There's a nucleus, Golgi apparatus. We have some vesicles. We don't know what they are, but some sort of vesicle. more vesicles, nucleus, vesicles, more vesicles. All right. Let's see here. Looking at this image, what would this be called?
Little walking guy. Walking guy.
Oh, is it things that walk to things?
Yeah, it's like the one with the.
Yeah, it's the walk. Prometheus.
Always one second.
Yo, this is the phospholipid bilayer. This is the plasma membrane. This, right, we're asking about the cell membrane. Yes, that is a plasma membrane, which is a phospholipid bilayer. It does kind of look like those proteins, the kinesins and the dyneins that walk up and down microtubules, right? But since there's two layers faced to the eye, you would never find motor proteins, right, all clustered together like that. These instead, right, are the phosphate heads, right, and the fatty acid tails. Right? This red protein right here, what generic name might we give it? Yeah, right, because it's integrated all the way through the membrane, right? So we have a cytoplasmic side and an extracellular side. What about this one? That's actually probably also integral, but what if it was just associated with the outer leaflet of that plasma membrane? Then what would we call it generically? We have integral proteins and we have peripheral proteins. Yes, very good. What's an example of a peripheral protein that we talked about just on Monday even? Tuesday, I mean. What's today, Thursday? Yeah, Tuesday. What's that? That's not a protein. Anybody think of an example of a peripheral protein that we talked about on Tuesday? What did we talk about on Tuesday? Can anybody tell me what we talked about on Tuesday? We talked about cell communication, we talked about receptors. The first type of receptor was what? G protein coupled receptor. And was that G protein inside or outside of the cell? It was inside the cell. And it was associated with the membrane. We'll look again here in a second. Yeah, a G protein is an example of a peripheral protein because it's associated with just the inner leaflet of the cell membrane. What do you think these three images depict? Yeah, cholesterol, good, right? You might not be able to tell from this or this, but here you can see the four fused rings. That's characteristic of a sterol molecule or a steroid molecule like cholesterol. Right? Cholesterol, right, could be embedded in the membrane. It wouldn't go all the way through. It would be embedded in one leaflet or the other. What is one reason that we might find cholesterol embedded in the membrane? Yeah, right. Some combination of what you're both saying, right? It decreases the fluidity of the membrane, right? Thereby stiffening or stabilizing that area of the membrane so that proteins that we need to be closely associated with each other, right, don't go floating away from each other because the area of the membrane that they're embedded in, right, is stiffer and less fluid, and so they're more likely to stay in place so they can interact with each other. What do we think this thing is? I know it's kind of a fuzzy image. This is not the best image I could use for this question, but maybe you'll get it anyway. What's this thing down here? This is definitely a protein, right? We can see the alpha helices. Right? If we see alpha helices, we know we're looking at a protein because that's one of the structures of protein. So what might be this thing that's stuck to the protein? There we go. It's a glycocalyx. If it were a less fuzzy image, right, you would recognize the glucose molecules, right? I know it's a little fuzzy, so maybe you can't see the glucose molecules, but if I showed you a higher resolution image where you could make out those glucose molecules, that should trigger you to think, what happens when carbohydrates are stuck to proteins? That's a glycocalyx, and so we would call this a glycoprotein, right? If instead, right, I had one of these lipids over here with the same glycocalyx stuck to it, we would call that a glycolipid, and that is a common, right, modification of proteins and lipids that are part of cell membranes. What are 3 ways that cells can regulate traffic across the plasma membrane? How do cells control what enters and leaves the cell? Hear whispers of things that are not wrong. Can anything diffuse across the plasma membrane? Why not? It is selectively permeable. Why? That's another answer. Yeah, it's got a fatty acid core, right? The plasma membrane has phosphates on the outside and inside, but the middle portion is all lipid, right? So what probably cannot diffuse through the plasma membrane? Anything that's polar. Anything at all that's polar, right? Because there's nothing, right, with those fatty acid tails that a polar molecule can associate with. So how would a polar molecule even get in there in the first place, right? So yes, right, by simply having a phospholipid bilayer, right, they can control, right, whether or not polar molecules can move across there, right? What about size, right? Even if it was a non-polar molecule, right, we have some size exclusion just because the phospholipid fills that space. How might we get a large molecule across the lipid membrane? Yeah, we could do some sort of transport molecule. For example, an aquaporin, right, is a pore that is the right shape and size for water molecules to pass through. Let's see here, and then Evan mentioned concentration gradients, right? Cells also spend a lot of energy pumping things out or pumping things in to maintain specific concentrations. So for example, right, they pump sodium out, right, and they pump potassium in. And so if given the chance, right, sodium wants to move into the cell because it's going to move down its concentration gradient because the cell works very hard to make sure that there's not a lot of sodium inside in the first place, and vice versa. Right? So by maintaining different concentrations of things inside and outside the cell, right, by having a phospholipid bilayer in the first place, which provides resistance to polar molecules, right, as well as very large bulky molecules, and then by creating channels, right, for specific molecules that it might want to move into or out of the cell. Those are 3 potential examples for how cells regulate that traffic. Let's see here. What are these 3, well, collectively, what are all these called? There we go, endocytosis, right? Because things are moving into the cell. All right, what's this first one? You're right, phagocytosis, right? We're creating a vesicle to surround a very large thing, like another cell, a microbe, a very large nutrient molecule perhaps, right? What about this one? That's pinocytosis, right? Taking little sips of ions and other small molecules there by creating a vesicle again. What's the last one? Come on, the last one's the easiest one. Right, can you see these little red things here? What do you think those little red things are? Yeah, those are receptors, right? So this is receptor-mediated endocytosis. Right? So pinocytosis, right, it just makes a vesicle and whatever happens to be there gets pulled inside. And receptor-mediated endocytosis, right, only the things that stimulate those receptors get pulled inside. What's similar between pino and receptor-mediated endocytosis? The vesicle is coated with coat proteins, right? Yeah, so in both these cases, right, we have coat proteins that help form that vesicle. Let's see here. What are some examples of molecules that can simply diffuse across the plasma membrane? Yeah, small lipids can. They're also nonpolar, right? We mentioned on Tuesday gases, right? Gases are typically very small molecules also that can diffuse across there, also nonpolar. Let's see here. Yeah, by what transport vehicle do contents destined for exocytosis travel to the plasma membrane. How would they get there? All right, the Golgi apparatus is the structure that they'll pass through that creates the transport vehicle, but what's the actual transport vehicle? What is it that's going to fuse with the plasma membrane? Yeah, it's just a vesicle. Let's see here. Passive transport includes what? Two mechanisms? Diffusion, which we've been talking about, and facilitated diffusion, right? Those are both diffusion, right? So one of them requires some sort of protein channel, something like that. The other one doesn't, but we're still moving down our concentration gradient, so it doesn't require any kind of energy. What are 3 examples of active transports? Co-transport was one. An ion pump was one. It's just a general mechanism. Trying to think of the name of it. It's escaping me at the moment. All right, but that sodium potassium pump was the other one. So I guess that's another type of ion pump. Maybe we had 2 examples. We have ion pumps. We had a couple of variants of ion pumps though. We had the sodium potassium pump, and then we had the proton pump, right? And then we had a co-transporter that used the gradient of protons, right, to co-transport something as well. 3 general types of cellular signaling, thinking very generally here. What's a long distance type of cellular signaling? We just covered this, right? So we haven't had a chance to read and study it. I get it. Right, so we had hormones, right? Hormones are long distance because hormones are typically released from the pituitary gland and then travel through the blood to perhaps a distant site to stimulate a cell that has a receptor for that hormone. Right, so what about close contact? 2 types of short distance signaling. What's that? So we just have junctions, right? Things like gap junctions, right? So when we have cell-cell contact, either through ligand and receptor, right, when they form that junction, or a gap junction that goes all the way through from one to the other. Right, so we have that very close contact, right, that requires contact of the cells. What if the cells are near each other but not actually contacting each other? What do we call that one? Call that one paracrine. Right? Paracrine is when a cell releases a signaling molecule into the extracellular environment and a nearby cell is stimulated by that. So we have cell-cell contact, right, through those junctions. We have paracrine when they're close but not necessarily contacting each other. Then we have, for example, hormones, which can be long distance. 3 types of signal reception here, right? So now we're talking about receptors. That's also something we covered on Tuesday. Which one do we think this one is? It is an ion channel. We have a couple of different types of ion channels. Even more general than that. Using the word gated. There are 2 different ways we can open an ion channel. The ligand gated, right? That's what this one is. This one's ligand gated, right? Because we have some sort of ligand that's stimulating the receptor that opens it, that allows ions to flow down their concentration gradient. What's the other way we can open an ion channel? Voltage. Right? So we can have ligand and voltage gated ion channels. Right? What about that second one? What is that? RTK stands for, talking about receptors, so that's probably what the R stands for. Right, so an RTK is a receptor. The relevant amino acid is tyrosine, right? So a receptor, tyrosine, and then what is something that can add a phosphate to something else called? It starts with a K, since that's the letter we're looking for. Kinase, right? A kinase is an enzyme that transfers a phosphate. So RTK, receptor, tyrosine, kinase. What is this first step here, right? Binding of the ligand leads to? Not yet. All right, we start out with two monomers which form a, it's not monomer, but a dimer, yes, right? So the first step, right? Ligand binding leads to dimerization, right, of the formation of a dimer. Then what happens? Okay, we just covered it on Tuesday, right? Then the tyrosine regions, right, autophosphorylate each other, right? So the two monomers, right, have all these tyrosines. The tyrosines are going to facilitate the phosphorylation of each other, so they autophosphorylate, right? Then what could happen once they're fully phosphorylated? Some enzyme could come along, right? Some enzyme that needs to be activated by phosphorylation, right? And so that enzyme comes along, right? Here are phosphates, right? That phosphate can be transferred, thereby activating that enzyme. And that enzyme, now that it's activated, right, can lead to a whole phosphorylation cascade downstream of that. Right? That's the receptor tyrosine kinase. What's this one over here? Yeah, right, otherwise known as a GPCR, which stands for a G protein coupled receptor, right? We can recognize it's a G protein coupled receptor because, right, of the 7 transdomain regions, right? So 1, 2, 3, 4, 5, 6, 7 alpha helices, right? So this protein goes through the membrane 7 different times with an alpha helix, which is why GPCRs are very often called 7 transmembrane domain proteins, right? But when we see those 7 alpha helices, right, we can identify it as a G protein coupled receptor, right? Again, ligand binding leads to what? What happens when the ligand binds a GPCR? It does activate it, yeah. All right. What about the shape of the protein? Does it change? Always. Right? When you bind something to something else, it's going to change shape, 100%. Right? So ligand binding leads to a conformational change of this GPCR, right? That, right, causes something to open up here that allows that GDP-bound G protein to bind to it. Right? So the G protein, when it's inactive and bound by GDP, doesn't have a spot to fit on there, right? Until the receptor gets activated, then, right, that's going to cause a twist in the shape that reveals that binding spot for that G protein. That G protein is going to bind. The GPCR then facilitates the binding of GTP, right, rather than GDP. Now that G protein is activated, GTP bound, activated, right? It's going to move along the plasma membrane until it runs into an enzyme that it can activate, something like a phospholipase, for instance. That phospholipase then, right, will be activated by that active GTP. Right? At some point, that GTP will auto-hydrolyze, right, that GTP, and go back to being an inactive GDP-bound version. And then whatever it activated, right, is going to start another phosphorylation cascade. 2 really similar mechanisms there. Almost done. In fact, this is the last one. What are some examples of second messengers? Yeah, I heard cyclic. Cyclic what? If we're rich with energy, what do we have a lot of? Yeah, right. Rich with energy is ATP. Poor in energy is AMP, and AMP can cyclize, right? It becomes cyclic AMP, right? So cyclic AMP is the second messenger. What's another one? calcium ion, right? Calcium's the second messenger. And what's our third example? It's already on there, right? The other one is IP3. What does IP3 stand for? It's inositol triphosphate, right? Remember I just had, I had several things listed. I just erased it, right? I had serine, choline, inositol, ethanolamine. Those were all examples of that nitrogen-containing group that's attached to phospholipids sometimes, right? So that's where the I comes from, inositol. So it comes from a phosphatidyl inositol specifically. Inositol triphosphate. And where does that IP3 and the, what does DAG stand for? Okay, again, this is just stuff that we talked about on Tuesday, right? Diacylglycerol, right? Let's make sure this makes sense to us. I shouldn't have erased it because it's still relevant. Right, so there is an example of a membrane phospholipid. In this case, the one that's relevant because it's phosphatidyl inositol, right? A phospholipid with inositol bond to it, right? So that's phosphatidyl inositol, right? If the appropriate enzyme comes along, can cut that, right? And so here is our inositol, right? I know I've only got one phosphate drawn there, but in reality, it's inositol triphosphate, right? So that's where the IP3 comes from. And what's the rest of this thing called? Right, well, it's two acyl groups or two fatty acids attached to glycerol, so that's diacylglycerol, right? So this is your diacylglycerol, and that's your inositol phosphate up there, right? So, what's the answer to the question? Where do IP3 and DAG reside in the cell before being activated as second messengers? Now, they're just components of the phospholipid bilayer, which I think is incredible. It's the most interesting thing we've talked about so far, right? Is the fact that we can take the phospholipid bilayer, which is a structural thing, and turn it into a signaling molecule. Yeah. How do we feel about that? I mean, not that great because you didn't know the